AP Biology. Today I'd like to talk about population genetics and microevolution. So, I'd like you to consider a gene locus that has two alleles, a big A and a little a, and if you figured out all the possible Punnett squares that you could draw, they're shown right here. And what I'd like you to do is stop the video and stare at this screen for a little bit until you see a pattern. So the pattern you should come up with is that all the top left-hand side are big A, and the bottom right-hand side are little a, and then the other two quadrants are big A, little a. And so this entire population really can be represented by a single Punnett square of a cross between two heterozygotes. So let's imagine that you could go into the population and you could see each allele that's around. You could go into each individual and count how many big A's are in the population and how many little A's are in the population. And you could probably do that with PCR and electrophoresis. And let's say you found that the frequency of big A was 0.3, or 30% of the alleles were big A. What would the frequency of little a be? Well, if those are the only two in there, it should be 0.7, because they have to add up to 1. 70%, 30% have to add up to 100%. Okay, so if we say that the frequency of big A, we call that P, and the frequency of little a, we call that Q, then P plus Q equals 1. All right, then let's figure out what would be the frequency of getting two big A's together to make a homozygous dominant genotype. So hopefully you figured out that it should be 0.3 times 0.3, or 0.09. Realize a lot of people make the mistake that's not 0.9, which would be 90%, it's 0.09. So again, if we say the frequency of big A is P, then the frequency of big A is going to be P squared. Okay, then what would be the frequency of getting two little a's together to make little a? Well, hopefully you came up with that it would be 0.7 times 0.7, or 0.49. And so here, if we make the frequency of little a be Q, then this is Q squared. All right, then imagine you had a heterozygote, and realize there are two ways of making a heterozygote. You can get a big A from mom and a little a from dad, or you can get a little a from mom and a big A from dad. So there are two ways to do that. All right, so you should figure out that either way you do it, it's 0.21, or 21%. And since there are two ways to get them, then you have to add the probabilities together. Or the frequency is going to be 2 times 0.3 times 0.7, or 0.42. And again, if we make, big A be, frequency of big A be P, and frequency of little a be Q, then this is 2PQ. And so, I'd like you to take a look at these numbers that we have, 0.9, 0.49, and 0.42, and figure out what is the relationship between those. And hopefully you came up with the idea that those added up to be 1, and so we can add P and Q in, and have these be P squared plus 2PQ plus Q squared equals 1. Okay, so these are the genotypes in a diploid, and then P and Q are the allele frequencies. And so these are the two equations, P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. And you don't have to memorize these for AP Bio since they're on the equations and formulas sheet. But these two equations describe the Hardy-Weinberg Equilibrium. There are, if you had more alleles, there are more complex versions of this, but this describes the Hardy-Weinberg Equilibrium, and it predicts allele and genotype frequencies in a population that isn't evolving. Okay, now we're going to talk later about things that the assumptions of the Hardy-Weinberg equilibrium, which when they're violated, and they usually are in nature, you get evolving populations. But this is an important thing because this is our null hypothesis for microevolution. So if we have allele frequencies from a real population, we can compare them against the predictions of these equations and see if the population is in fact evolving.